Hello. Yeah. Okay.
Participants can kindly join the meeting now.
All right. Welcome back. I have two talks on anaerobic digestion today. First talk is on some of the advances in this process. We mostly concentrate on waste to energy, different wastewater and bioreactor, and how to use anaerobic digestion to make energy in the form of methane and hydrogen. Then the afternoon we talk about biogas and alternative use other than methane, what else we can produce, okay? See, is it not moving? Can you make it move? Yeah. All right. So if you look at the waste we produce in the world, 90% of the world's wastewater does not receive any treatment at all. So it just goes right into the environment. So only 10% of the waste we produce in the whole world is go through some kind of treatment, go through treatment process. And one other method we use anaerobic digestion as a way of treatment. Okay, so most of the waste is not treated at all. So we're going into the environment. What's going on? So let's look at some of the biological treatment systems. So some of the terms you should know. I know most of you know, but I'm going to go through. You know what is BOD, COD, and solids, and these are the terms we use in wastewater treatment process. Whoops, it's going too fast. So BOD stands for biological or biochemical oxygen demand. It's defined as the amount of dissolved oxygen needed by aerobic bacteria or aerobic decomposers to break the organic material in a given volume of water at a certain temperature over a certain period of time. So they do call BOD 5 or BOD 3, how many days you incubate to measure that. So BOD is a measurement of biologically degradable organic matter. Okay, so carbon, most of them can be degraded, but there are some carbon cannot be degraded, like carbonates. So we are talking about BOD, only those carbon can be degraded. So how much oxygen is needed by aerobic bacteria to break down organic carbon is called BOD. That is one of the common measurement to find out pollution level in the water. Okay. COD on the other hand is chemical oxygen demand. Chemical oxygen demand is the measurement of chemically oxidizable compounds, organic carbon in the water. This includes both biodegradable and non-biodegradable carbon in the water. Okay. So as a rule of thumb, there is more COD than BOD, right? There are two kinds of COD. You measure total COD that includes all the carbon that is dissolved in the liquid and also the biomass, which is bacteria, algae, fungi. The carbon comes from those cells. And when you separate the biomass and only measure carbon in the liquid, and that is called soluble COD. Yeah. Oh, okay. Yeah, okay. So there are total COD and soluble COD. Total COD includes everything, including the microbes living in the water. Soluble COD measures only the dissolved organic carbon. You separate the biomass out of the liquid. Okay. Can you move it for me? It's not moving now. Okay. The other term we use in wastewater is solids. How much solids are there? We have total solids, suspended solids, dissolved solids, volatile suspended solids. Okay, those are different solids measurement that we use for loading the reactor, and then measuring volatile suspended solid, measure the biomass in the reactor, how much bacteria are there. We can, as a crude way of measuring volatile suspended solid equal to how much microbes in the water. Okay. So the definition is there. That total solid is all the particles that remain after evaporation at 105 degree as a residue. Suspended solid is the particles that are suspended in water. You go through filtration and see whatever is suspended. And dissolved solid is all the ions that is dissolved in the water. Okay, that's the dissolved solid. And then we use reactor is a vessel or a container where you put the wastewater in there to make the microbe work on the waste to remove the carbon, and that is we call reactor. Then we use a term called organic loading. The organic loading is the amount of carbon or amount of solids that we put in the bioreactor. Okay, that's what these terms are referred to when you talk about wastewater treatment. So in biological treatment systems, basically the three processes, physical, biological and chemical, which is called primary, secondary and tertiary treatment. So primary is basically physical process. You screen the wastewater and remove anything that is floating in the wastewater, and then you use the settling tank to settle the solids out. So you're using the physical process to remove the debris, remove the solids. That's called physical process or primary treatment. The secondary treatment is biological. That's where you use microorganisms to break down the carbon and reduce your BOD, COD in the water. Okay, that's where you play with the microbes reaction to achieve 90% removal of carbon, or based on how effective your bioreactor is. Okay. Common one is activated sludge, which is basically you aerate the wastewater, because the limiting step is oxygen. So when you provide oxygen, the aerobic bacteria will multiply and reduce the carbon, convert them to biomass and carbon dioxide. The other one is trickling filter, commonly used in sewage treatment system. And the tertiary system basically is a chemical process. You disinfect the wastewater before you let them out in the moving water body. Usually chlorination, we use ozone, UV treatment, a lot of different way to use tertiary treatment using process to remove pathogens. The other terms you need to know is loading rate, how much carbon is going into the system, how much solid is going into the system. Flow rate, how much volume of water per day or per hour is going in there, that's flow rate. Hydraulic retention time is commonly known as HRT, which is the time of wastewater, the time that's spent inside the reactor, whether it is 1 day or 2 days, you're reacting with the microbes. That is called HRT. The amount of time the wastewater spends in the reactor is called hydraulic retention time. SRT stands for either sludge retention time or solid retention time. This refers to how much time the microbes are inside the reactor. So when you continuously pump in the water into the bioreactor, you tend to lose microorganism, right? It comes out on the effluent. The influent pumps in, effluent comes out. Some of the microbes will leave, right? Your job is to keep your bacteria as long as possible. So when you design a reactor, you have to design a reactor that has long SRT and short HRT. You want to continuously pump water and remove the carbon, so you don't want to hold your water long time. But at the same time, you want to hold your bacteria a long time. You don't want to lose the bacteria. So that's a major criteria in designing bioreactor. You need to have long SRT and short HRT, hydraulic retention time. Okay, that's the basic rule of designing bioreactor. Okay, then you need to process analysis when you deal with wastewater to energy system. How much carbon is going in, how much carbon is converted to whether it's biomass or methane or carbon dioxide. So you analyze what is going in the reactor, what is coming out the reactor. You work out the mass balance of the system. That is called process analysis. And you also work out the mass balance of either carbon, mass balance of bacteria, everything you can work out. So these are some common terms we use in wastewater when we deal with wastewater to energy or wastewater treatment itself. You should know that. Okay. So let's talk about anaerobic digestion. That's what this lecture is about. So the anaerobic digester is a big vessel, and it operates under anaerobic conditions. So you have no oxygen at all. So you operate under anaerobic condition. So you operate under a certain temperature to allow the particular group of bacteria that does the ultimate final oxidation step, which is methanogenic bacteria. Okay, so you operate them in steady state. So the end product you get from wastewater treatment by using anaerobic digester is the energy, the gaseous fuel, in this case methane. So most of the time there's a general composition of your gas is 55 to 65% methane. The bacteria convert carbon to methane gas, and 35 to 45% is carbon dioxide, and then you also produce hydrogen sulfide, nitrous oxide, nitrogen. Other products are also being made in the bioreactor. So the bacteria does the simple thing. They break down your carbon and produce these gas, and see the methane is your natural gas, and that's what we can use it to, for your cooking purpose, or you can also convert them to electricity. You can use a generator and produce electricity. So your end goal is to break a carbon from the waste and produce this gaseous fuel, okay, methane. So let's look at the steps involved in anaerobic digestion, how bacteria break it down. Generally, there are only 4 processes involved in the digester. First step is hydrolysis. Second step is fermentation. Then you have acetogenesis and then methanogenesis. The hydrolysis is converting complex polymer into simple monomer. Okay? So when you look at the wastewater, basically it's carbohydrate, protein, and lipid. Carbohydrates can be converted to sugar, and protein is converted to amino acids, simple monomer, and lipid is converted to fatty acid and glycerol, right? So that's what that first step is. The group of bacteria, we call it hydrolytic bacteria, carry out hydrolysis reaction. So it can break down carbohydrate to simple sugar, it can break down protein to amino acid, break down lipids to fatty acids, okay? That's what's going on in the first step, okay? The second step, the group of bacteria, they ferment because an anaerobic condition. So what they do, they ferment this monomeric compound and produce fatty acids. We call acidogenesis or fermentation. And the fatty acids are different fatty acids, produce acetic acid, butyric acid, propionic acid, valeric acid, isobutyric acid. So these are all short chain fatty acids. Okay, a lot of fatty acids produced. That's your second process. And when the fatty acids are produced, then we call acetogenesis, which is a group of bacteria that take that other fatty acids, convert them to one fatty acid, that is acetic acid. That is a favorable acid for microorganisms. Okay, that process is called acetogenesis. Okay, that's the third step in anaerobic digestion. And the last step is the final terminal oxidation step. Methanogens take over the acetic acid, and also hydrogen and CO2 produce, convert them to methane and carbon dioxide. So that completes the anaerobic digestion process. So the important thing to remember is 4 chemical reaction take place is hydrolysis. Fermentation we also call acidogenesis, produce all the acids. Okay, then convert the acids to one acidic acid that's called acetogenesis, and then we have methanogenesis. So if you look at it, there's a complex biodiversity of microorganisms. There's involved several thousands of bacteria of different species. They have to cooperate and coexist, and if one species dominate, the whole thing will collapse. All right, so you have to work in concert to achieve production of your final product of methane gas. So let's look at the go back one more step. So limiting step here. Go one more slide. Yeah. So this is not a complete conversion of organic matter. They produce digestate. So whatever left after the gas is produced, you produce digestate. The biogas is not. We call it biogas, but it not only one methane. It also has other gases. As I said before, it has carbon dioxide, it has hydrogen sulfide, and all the other gases in there. So if we put in a simple perspective how anaerobic digestion works, is a big bioreactor operated anaerobically without oxygen. So what is the input going into the system? You have the organic carbon. It could be carbon from wastewater, carbon from different feedstock. Then it has nitrogen, phosphorus, sulfur, ammonia, phosphate, anything that's in your wastewater going into this digester. And then you have this all four different processes we talked about work inside in concert, and it produces variety of gases. Mostly it is methane. Okay, and then the liquid output contains the digestate also contains nitrogen, phosphate, residual organic carbon, and hydrogen sulfide. And these are really good, rich in nutrients, so it could be used as a biofertilizer in the farm. It's really good fertilizer. It's really a lot of stuff. And there is some spent solids that come out we call sludge, and sludge can be processed and reused as a soil amendment back into the farm. Okay. So let's talk about the process, how it works. So microbiology behind it and environmental factors to make this work in really optimum conditions. All right, so if you look at all these four stages we talked about, we have different reaction takes place in these four stages. We have complex polymer, polysaccharides, protein, fat, are converted to simple monomer as sugar and amino acids, and then you have intermediate product that produced that is converted to acetate and short chain fatty acid, and then we produce not only acetic acid, you also produce hydrogen and carbon dioxide. And there are two kinds of methanogen. We have acetogenic methanogen, hydrogenotrophic methanogen. Acetogenic methanogen take acetic acid, convert them to methane and carbon dioxide. Hydrogenotrophic methanogen take hydrogen and CO2 and make methane. Okay, so if you look at it, you have last slide I showed you 4 processes. Now I'm showing you 7 processes. Okay, there is a lot of reaction going on. So you have hydrolysis, acidogenesis, acetogenesis. Those are clear, right? Then we have homoacetogenesis. You have syntrophic acetate oxidation. And then we have acetoclastic methanogen, which is the bacteria that take acetate to make methane. Then we have hydrogenotrophic methanogen that take hydrogen CO2 to make methane. So we have a whole lot of other reaction takes place inside, and that's what the number is showing in this slide. You have 1, 2, 3, 4, 5, 6, 7. All those are where these bacteria play a role in this chemical reaction inside. Okay, so a lot of things going on inside the digester. Can you move? Okay. The rate limiting step for complex substrates such as lignocellulosic, when you are using co-digestion, biomass or sewage sludge or animal manure is your first step. Okay. When you are using complex feedstock like plant material, like a plant material, I told you need pre-treatment, right? So the hydrolysis step, you can use plant material directly into the digester, but it has to be some kind of co-digestion. It has to be with manure in a ratio. Okay, that's your rate limiting step for the feedstock that are lignocellulose. And then the next slide. Okay, we look at the rate limiting step for the weak material, like readily degradable substrate, are the last step. So when the carbon can be readily degradable, the hydrolysis reaction is fast. As a result, your system accumulates a lot of fatty acids. Okay, and the fatty acid, when it accumulates, then it's going to inhibit your next methanogenic bacteria. So the methanogen will shut down. So depends on your feedstock, the rate limiting step could be hydrolysis or your osteoclastic adrenotrophic methanogen, okay, based on what is your substrate in your system. Okay. So this is just to put together the same information in the same slide. Next slide here, it shows hydrolysis, fermentation, syntrophic acidogen, and syntrophic acetate oxidation, homoacetogenesis, all of them, and showing you reaction where it takes place and what we call them by name in anaerobic digestion process. That's a simplified system of explaining how anaerobic digestion works. Okay. So let's look at process microbiology. So anaerobic degradation carried out by series of microbes. Okay, so certain microbes work with first set of reaction, produce a product, and then another set of microbes take that product and make another product. So it's a series of metabolic pathway. Different organisms work in different chemical reactions. So it has to be in a coordinated interaction. If one system produces more than the other, the thing will collapse. Okay, the process may fail if certain groups of microbes are inhibited, or certain groups of microbes are overactive. Okay, accumulate fatty acid, accumulate hydrogen sulfide. A lot of other problems will come into play. So let's talk about process microbiology. So fermentative microbes, this group of microbes respond for the first stage, hydrolysis and acidogenesis collectively called fermentative microbes. These bacteria are, the first step is facultative microbes. So they can tolerate oxygen. They can grow in the presence and the absence of oxygen. Those are called facultative microbes. And there are also some strict anaerobes that play a role in that first step. And then we have hydrolytic bacteria that decompose insoluble organic compound to soluble form by making exoenzymes, and that group are present in the hydrolytic bacteria. And then we have the bacteria that are actually doing the acidogenic phases that have energy for growth and reproduction. That's the next step in the bacteria that involve. So some of the common bacteria that does is the family of Streptococcaceae and Enterobacteriaceae in your hydrolytic reactions, the first step. And then we have other genus present in this, the name of the bacteria listed here, Bacteroides, Clostridium, Vibrio, Eubacterium, Bifidobacterium, and Lactobacillus. All of them can work in that concept in hydrolytic reaction. So I'm throwing you some names, but these are the ones commonly carry out the reaction on hydrolytic step. Okay. The hydrolytic bacteria with the corresponding enzyme, I show you name of the bacteria, what it does. Okay. So it produces those exoenzymes, and those are bacteria that produce that, and the bacteria that work on the substrate, and then what product they make. For example, if you look at polysaccharides, that could be cellulose, starch, or pectin, mostly in your plant material. The enzyme you need to break it down is cellulase, amylases, and pectinases enzyme. The bacteria that produce these enzymes are Clostridium, Bacillus, Lactobacillus. So the exoenzymes, they chop those polymer into monomer. Okay? As a result, you produce simple sugar, okay? Glucose, xylose, whatever. Okay? And then the substrate is rich in protein. Example, it's a slaughterhouse waste, or there's a lot of fishery waste. It has a lot of protein in it. And you need an enzyme called proteinase and peptidase enzyme to break down the protein. The bacteria that produce these enzymes are listed here. These are Bacteroides, Butyvibrio, Clostridium, Fusobacterium, Selenomonas, and Streptococcus. And these bacteria produce these enzymes, break down the protein to make simple amino acids. That's a monomeric compound. And then the last one is the fat. If there is fat-rich substrate, the enzyme you need is lipase. Lipase producing bacteria are Mycobacterium and Clostridium. And these bacteria can produce lipase and break the lipids to fatty acids and glycerol. Okay? So these are the name of the bacteria, the name of the exoenzyme, the final product from your substrate. So your substrate could be rich in carbohydrate, could be rich in protein, or could be rich in fat, depending on which wastewater you're treating, okay, where it comes from. Right? Your effluent could be food industry, or leather industry, or aquaculture industry. So depending on what kind of waste, you could be rich in protein, a lot of nitrogen in it, rich in carbohydrate, or rich in fat. So your starting material is different, so you have to have different combinations. So the beauty of the system is you have all these bacteria in your digester. You don't have to add bacteria. The bacteria will, in the presence of this material, they will increase in population, produce an enzyme, break down for you to simple monomeric compound. Okay? Let's look at acidogenic bacteria. The acidogenic bacteria metabolize organic acids, C2 compounds, and alcohol and certain aromatic compound like benzoate or benzene into acetate and carbon dioxide. That's a reaction of the acidogenic group of bacteria. So that's a reaction show you from propionate to acetate, how bacteria break down propionate and produce acetate and carbon dioxide and hydrogen. And the bacteria that carry out in your system is listed here, the Syntrophobacters, propionate to acetate, they do that. Syntropomonas bacteria that carry the butyrate with one more extra carbon, a longer chain fatty acid into acetate, they break it down. And then we have syntrophic association, hydrogen producing acetogens, they can produce hydrogen. And then you need to, there is a problem when you have excess hydrogen production, the propionate degrading bacteria are going to be inhibited. So you need to make that interspecies hydrogen transfer balanced there. So if you don't want to produce too much hydrogen in the system, that will inhibit other group of bacteria. As I said here, the propionate degrading bacteria is suppressed by excess hydrogen in your system. So in order for this process to work, all these bacteria should work in concert, in coordination. Otherwise, they will have some system imbalance. Homoacetogens are a group of bacteria, and they are either autotroph or heterotrophs. Heterotrophic homoacetogens break down organic substrate, example like formate, methanol, and convert them to acetate. So acetate is your key. Everything is converted back to acetic acid in the system. Autotrophic homoacetogen take hydrogen and CO2 and make acetate. The reaction is given here, the 4 molecules of hydrogen and combined with carbonate, and you produce your acetate, and that's a favorable reaction. So that's what the autotrophic acetogen can do, homoacetogen can do to make acetate. So there are 2 ways to make your acetate, from organic compound and from gaseous compound, inorganic. And then we have energy release. When hydrogen is used, hydrogenotrophic organisms release energy, and hydrogen oxidation, homoacetogens have energy release. Okay? So there's a competition between hydrogenotroph and homoacetogens for available of hydrogen. So the hydrogen is a key balance of accumulation of hydrogen. So the bacteria has to take the hydrogen out. So the autotroph homoacetogen will do that for you in the system. Okay, so you don't have excess accumulation of hydrogen. So at low hydrogen partial pressure, hydrogenotroph dominate over homoacetogen. So Clostridium acidicum and Acidobacterium woodii are home acidogen commonly present in sewage. Okay, again, all these bacteria are there. Again, you don't have to add anything. The bacteria present in the sewage anaerobic digester. Any anaerobic digester, you take sample from, you find these bacteria. Okay. So let's look at the syntrophic acetate oxidizing bacteria. What are those? These are acetate oxidizing bacteria, exist in syntrophic association with hydrogenotroph. They work in concert with two groups. Okay, that's why it's called syntroph. Syntrophism is two organisms working together. So acetate is oxidized to carbon dioxide, producing hydrogen by the syntroph acetate oxidizing bacteria, and it's thermodynamically driven by your hydrogen consumption and by the hydrogenotroph. Okay, that's the chemical reaction shown in here. So you have the acetate combined with your water and hydrogen, and water with the water, and you produce your hydrogen. Okay, that's your bacteria that carries out production of hydrogen. And then you take the hydrogen combined with carbonate and you make your methane. Okay, so methanogen will take that hydrogen and you make your methane product. So at high concentration of inhibitors, if you have too much ammonia and volatile fatty acids, accumulation of fatty acid, that will inhibit the methanogenic bacteria. So that's why you need this syntrophic association. These two groups of bacteria has to work in concert. Otherwise, there's going to be the imbalance of overproduction of acetate, overproduction of hydrogen will make this system upset. Okay. Next slide. The last group of bacteria that carry out production of methane gas is your methanogenic bacteria. And the methanogenic bacteria are not true bacteria. They are called archaea, as an ancient bacteria, and they are distinguished from true bacteria because it has different cell membrane, different type of cell wall. They don't have peptidoglycan like all other bacteria. It has a different ribosomal RNA, they're totally different domain than bacteria. Okay, and these organisms are obligate anaerobe. They don't like oxygen. It's completely anaerobic bacteria. Okay, and the rate limiting species in anaerobic treatment is rapidly changing substrate, degrading substrate. So that's your rate limiting step. If you have too much fatty acid, your methanogen will shut down. Okay, so methanogens coexist or compete with sulfur reducing bacteria. The sulfur reducing bacteria are already there in the wastewater, so they compete for the same substrate. So if you have too much sulfur reducing bacteria, your methane output will be less, you produce more hydrogen sulfide. Okay, so if there is sulfate in your wastewater, then SRB, the sulfur reducing bacteria, will take over. Okay, so that's your final step. It's all dictated by the redox potential. When redox potential is negative 400, methanogens is what dominating the system. Okay, all right. So those are various microbes in your digester break down your carbon and produce natural gas. That gas can be used as energy source. Okay. So the two main group of methanogens I said, there are two, one that break down acetate to methane, another that combine hydrogen and CO2 to make methane, and those are acetoclastic and hydrogenotrophic methanogens. Okay, next slide. So the acetotrophic and acetoclastic methanogens, these methanogens metabolize acetate directly into methane. Acetate is cleaved to methyl and carboxyl group. The methyl group is reduced to methane, carboxyl group is converted to carbon dioxide. So that's the two gas produced coming from methyl group or carboxyl group of your organic compound. The two genera of acetoclastic methanogen commonly present in your wastewater are the methanosayeda and methanosacina. The next slide, yeah, methanosacina. These are the two common ones. So methanosayeda is the rod-shaped filamentous low Ks bacteria. That means low affinity to substrate. It takes long time to multiply. it uses only acetate. It is able to grow at a low acetate concentration. The methanocysina are different structure. The bacteria is clusters. Okay, it's aggregate and it has high Ks value. That means it can multiply really faster. Okay, it has. It uses methanol, methylamine, and any kind of C1 compound and convert them to methane. It has a higher acetate threshold. It can take high concentration of acetate. So these are the two common methanogenic bacteria that present in anaerobic digester. Okay, next one. So this to show you what is the Ks value. Ks value is the K constant is the affinity of the organisms to the substrate. If it can reach half the maximum growth within short time, that is your Ks value. So here is the Ks value for methanocyaida and methanocysina. Methanocysina can multiply, if you look at the maximum growth, we call the mu, it reached half the mu in short time compared to methanocyaida. Okay? So it has a high affinity to acetate. They multiply faster. They reach half maximum growth within shorter time using shorter consumption of substrate. That's why we call high Ks, low Ks. It's called affinity to the carbon. Okay? So just to show you, these two bacteria are totally different. One has high Ks, another has a low Ks value. Let's look at the other group of methanogens that convert hydrogen and CO2 to methane. We call that hydrogenotrophic methanogen. They reduce carbon dioxide to methane. You put the electron into the carbon dioxide reduction process, you make your methane. Utilize hydrogen derived from catabolic reaction, you put hydrogen CO2 and you make your methane. The activity of this group of methanogen is crucial to control your hydrogen partial pressure. So hydrogenotrophic bacteria need to play their role to keep your hydrogen partial pressure low. If the hydrogen partial pressure is high, then your acid producing bacteria will shut down. Your propionic acid will accumulate over there. All right. So hydrogenotrophs can also convert other C1 compounds like formate or formic acid. That's a reaction there. Okay, next one. And the bacteria that convert the acetogenesis process all the other fatty acid to acetate, the reaction is given here. You have butyrate, propionate, ethanol. Bacteria can convert them to one fatty acid, acetic acid. And that's a reaction given. They all have favorable delta G. So values, so these reactions are favorable to carry out. That's why the bacteria play converting all the long chain fatty acid to simple fatty acid of acetic acid in your system. Okay, all right. There's another group called metallotrophic methanogens. They catabolize methyl group containing compounds such as methanol, methylated amine, methylated sulfide. They catabolize those type of compound in your waste and produce methane. The methyl group is transferred to a methyl carrier and reduced to methane. So those are the reaction is given here, whether it is a methylamine or methanol or anything else, the substrate, they are all converted to methane and carbon dioxide by this particular group of methanogen called metallotrophic methanogen. Okay. Let me see previous one. Okay, next one. Yeah, the next one is sulfate reducing bacteria. Next slide. Yeah, sulfate reducing bacteria are the bacteria that sometimes you don't want too much of it. As I said, it competes with methanogen, the same substrate, and your methane gas concentration will go down. So sulfate reducing bacteria also play a key role in in the anaerobic digestion process. So it coexists with methanogen, but they compete for same substrate, acetic acid. So sulfate is the electron acceptor. Okay, the bacteria use sulfate as electron acceptor, and electron that is generated from substrate is dumped onto the sulfate, and bacteria produce hydrogen sulfide that produce the rotten egg smell in your gas. So sulfide is reduced to hydrogen sulfide, which inhibits the growth of methanogen. If you have too much hydrogen sulfide, the methanogen population will go down in your system. So during anaerobic treatment of high sulfate containing wastewater, your sulfur reducing bacteria are going to outcompete your methanogen. Okay? So the kinetic and thermodynamic properties of SRBs is shown here. Right? They are competing for the same substrate. So bacteria take sulfur reducing bacteria take their hydrogen and put them on a sulfate, produce hydrogen sulfide. There is less hydrogen available for methanogen to make methane. They are competing in the same substrate. Okay? Sulfur reducing bacteria also take the acetic acid and convert them to hydrogen sulfide. Okay? Because they are dumping the electron into the sulfate. Your H2S is your end product when you have sulfur-reducing bacteria. The group of sulfur-reducing bacteria present in the digester are listed here. Those are D-sulfovibrio, D-sulfobulbus, D-sulfomaculum, D-sulfobacterium, D-sulfococcus. Those are all sulfate-reducing bacteria in your anaerobic digester. You don't want too much of it, but it is always there in the anaerobic digester. They compete for the same substrate. When there is too much H2S, you will have problem with methanogens. Okay, next one. So important consideration. So now we talked about all the microbial groups that carry out biochemical reaction in your digester. Environmental factors also play a key role in your digester performance, your efficiency of methane production. The successful operation of anaerobic reactor depends on environmental factors that are close and comfort to the microorganism involved in the process. Okay, those are common factors like temperature, pH, alkalinity, nutrient like trace elements. You need to have like iron, manganese, and all that. Then inhibitor. Sometimes there is inhibitor in your wastewater that will kill the microbes. And then how much loading rate, how much carbon going into the system, how much your SRT and the HRT we talked about, how much solid retention time in the system, how much hydraulic retention in the system. These all play a major role in your performance of your anaerobic digester. Okay, next slide. So let's talk about some of these factors. Environmental factors. The successful operation of anaerobic reactor depends on the temperature. Anaerobic process, like any other biological process, is temperature dependent. It has a range. Anaerobic bacteria, there are three kinds: cold-loving bacteria, hot-loving bacteria, and medium temperature bacteria. They are called psychrophile, mesophile, and thermophile. And we have the temperature range for these three group of bacteria. Psychrophile are cold-loving, anywhere from 5 to 15 degree. Mesophile like to grow anywhere from 35 to 40 degree, and thermophile like extreme hot temperature above 50 degrees centigrade. Okay, next slide. So if you look at this, this is the range, and graphically showing those three groups of bacteria. So anaerobic digester has all three groups, right? So depending on your external temperature where your digester is present, psychrophile will be active, a thermophile will be active. So most of the time it's mesophilic, but in cold countries like northern North America and Western Europe, there are psychrophilic system out there. Sometimes you need to maintain the mesophilic bacterial temperature. You need to put heating system in the digester. All right. Next slide. So the general rule of thumb, the rate of reaction doubles for every 10 degree rise in temperature up to the optimum range of the bacteria. Okay, so that's a general rule of thumb. It's every 10 degrees centigrade in the temperature will make a difference in your performance of reactor. Okay, let's look at pH, another major factor. Commonly, the acidogenic bacteria like to grow in the acidic pH range, 5.5 to 6.5. Methanogens like to grow near neutral pH, little bit alkaline pH, 7.8 to 8.2. And you can see the pH range the bacteria like to grow. Methanogens are more susceptible to pH change than other microbes. They are very finicky. You need to have that pH range for methanogens. So the operating pH for all the bacteria in general for the digester is 6.8 to 7.4. So that's why you need to have a buffering system. You have to have alkalinity in your liquid. Okay, so low pH reduces the activity of methanogens, causing accumulation of fatty acid. The hydrolytic reaction going to produce more acid. At higher partial pressure, when you have too much hydrogen, your propionic acid degrading bacteria will get inhibited. Your propionic acid will accumulate. It doesn't go to acetic acid. Okay, so when this kind of problem happens, we call that the sour in the system. It becomes stuck. The process not completed. We call sours. Okay, next one. So the remedial measure is to control your pH. So you may have to use some buffer. Try to bring the pH to that ideal range of 6.8 to 7.4. Okay. So naturally, these anaerobic digesters have built-in alkalinity. You don't have to add any buffer most of the time because it has all the substances, alkaline substances, to get your alkalinity, to get your pH maintained naturally. Okay. So anaerobic system has its own buffering capacities. Any pH drop they will try to maintain with that range. For example, when protein is degraded, ammonia is released, right? Ammonia, which reacts with carbon dioxide and produce ammonium carbonate. That's your alkalinity to adjust your buffer. When you have sulfate and sulfide reduction, you produce carbonate, and that's another alkalinity in your system. So these are some reaction bacteria carry out. So it has a natural buffering capacity. Okay, and extreme conditions you have to add alkalinity to adjust your pH. But most of the time, bacteria has this natural alkalinity production in your system. Okay, next. So other thing that you need is nutrient and trace element. Some of those combination we call that carbon-nitrogen ratio, carbon-phosphorus ratio, and then some trace elements bacteria need. Those are iron, manganese for your cofactor for enzyme function. Okay, so the macronutrient, our major one is your nitrogen, phosphorus, and sometimes sulfur, and then trace metals you need for your optimum enzyme function, which is nickel, cobalt, iron, molybdenum, selenium. Okay, so the general rule of thumb is if you are talking in terms of COD for carbon, the COD nitrogen phosphorus ratio for highly loaded system is for every 350 part of carbon you need 7 part of nitrogen, 1 part of phosphorus. That's called 350-7-1 ratio. If your system is lightly loaded, your loading rate is not high, your carbon nitrogen ratio, phosphorus ratio is 1000 7-1. For every 1000 part of carbon, you need 7 part of nitrogen and 1 part of phosphorus. Okay. So the nutrient requirements also, we call that CN ratio, carbon nitrogen ratio. Okay. Most of the time they are recommended in the ratio of, for anaerobic digestion, 20 to 30 ratio. For every carbon nitrogen, that range should be 20 to 30 CN ratio. The co-digestion of high carbon feedstock, if you are using lignocellulosic in your anaerobic digester, you need to have high nitrogen feedstock comes with it, so you can, you have no problem with that. Okay, it will achieve the proper CN ratio. Next slide. Then we have to consider inhibition. Toxicity come from your waste. Your waste has sometimes toxic material that will upset your reactor operation. So some of the common toxicity is ammonia. You have too much ammonia in your system. Heavy metals, and in the processing plant they use heavy metal halogenated compounds and cyanide, and those are all toxic to bacteria. So if that kind of waste is produced in the factory, then you need to do pre-treatment to eliminate this reduce the toxicity or dilute it. Okay. The other toxicity includes sulfide volatile ammonia, volatile fatty acid, too much volatile fatty acid. Okay. The other thing to consider is the relative distribution of ionized and non-ionized form to govern your operating pH. Alkaline pH favors more ionized form of sulfide, and acidic pH favor more ionized form of ammonia. So depending on what you have, is you're producing too much ammonia, too much hydrogen sulfide in your system. Okay. All right, then the next one is volumetric organic loading. How much carbon or how much solids going into your system? That you have to figure it out depending on your analysis of COD in your wastewater. The volumetric organic loading is simply we call OLR, is expression of mass of organic matter fed per unit reactor volume per unit time. Is based on either total solids or based on your carbon, COD. Is expressed as either kilogram of COD per cubic meter per day, that is you are inputting into the digester, or if it is expressing a solid, it's a kilogram of volatile solids per cubic meter per day. Right? You could do it either way, depending on your feedstock. Okay. The formula is given here. Volumetric organic loading depends on your substrate, how much COD is in your substrate, your wastewater flow rate, which is your Q, cubic meter per day, and your total volume of your bioreactor, cubic meter of bioreactor volume. So that's a formula. SQ divided by V will give you your organic loading rate, how much you wanted to put in there. If you measure your COD, you can calculate. If you know how much your bioreactor volume and how much COD is in your system, how much is flow going in every day, you can figure out how much carbon you want to put in your system. Okay. Next one. So the high rate of anaerobic reactors operated with organic loading rate of 10 to 40 kilograms COD per cubic meter per day. That's a commonly used loading rate. Organic loading rate for high solids, when you have a lot of solids, that rate is anywhere from 1 to 5 kilogram of volatile solids per cubic meter, which is 8 to 9 kilogram of volatile solids. Okay. So these are all already figured out. So if you have a specific type of wastewater, you can use this formula to figure out how much COD you want to put in your digester. Okay. Then the last thing is HRT and SRT calculation. You need to figure out how long you have to hold your bacteria, how long you have to hold your wastewater into the reactor. The HRT time for which the substrate remains in the bioreactor is, could be days, could be hour, could be, weeks. Your bioreactor, you're holding your waste. Solid reactor time, you always wanted to have more time bacteria. You don't want to lose your bacteria. We call that washout. You don't want to washing out of bacteria out of your system. You want to keep the good bacteria in the system. So you need to design your bioreactor to hold your bacteria inside for a long period of time. So we're going to show you some bioreactor design, how they do that. All right. So simple soluble substances are readily degradable substance that require short hydraulic retention time. If your substrate is simple, your hydraulic retention time is in hours, not in weeks. Okay, because bacteria is breaking down very fast. Okay, and if your complex substrates, if they're like a really lignocellulosic, as in your digester, that needs long HRT. Right. So depending on substrate, your HRT varies. Okay, and then your SRT based on your reactor, whether it is a continuous tank reactor or it is a filter system, you can hold your SRT longer by designing your reactor system. Okay, so you need to hold your bacteria long period of time. Okay, next one. So the other advantage of anaerobic digester, it also eliminates pathogens. So when you operate anaerobic digester, it reduces the pathogen in your wastewater, because anaerobic digester, under anaerobic condition, most bacteria die and they cannot compete with the anaerobic bacteria, and under the operating condition, your pathogens in the wastewater decreases. Okay, so the temperature and retention time of SRT also governs the pathogen in your system. Sometimes you need use lime treatment or sanitation before you get the sludge out if there are any pathogen in your system. Okay, so you can see the picture here. The green part is of the picture shows the way the manure, undigested manure applied because it's rich in nitrogen, phosphorus coming out of the digester compared to not applied. You don't see that many growth of plants. So the digestate comes out as a lot of rich in nutrient, okay, and less pathogen. Okay. So other essential condition for your digestive operation is to avoid excess oxygen. Sometimes the small amount of oxygen is good. I'll show you at the end of the presentation micro aerobic conditions. You need to make sure your toxicity is less. You don't have any cyanide or any other toxicity in your waste. Try to maintain your pH 6.8 to 7.2. That's a range you like it. Make sure you have sufficient alkalinity producing bacteria are there, so you don't have to add alkaline substances to control your pH. You have to have a low volatile fatty acid accumulation, temperature in optimum range, enough carbon nitrogen ratio, enough carbon phosphorus ratio, enough cofactors. All that we talked about is summarized here. Your SRT, HRT, find out for your particular wastewater to operate your condition. So these are all key factors. I talked about microbial part. I talked about environmental factor. Okay, let's move on to digester design to hold your SRT long time. So if you look at a reactor configuration, the simple system is a continuous CSTR, continuously stir tank reactor. You have the closed system, no oxygen. You have a big mixer to mix to have your mass transfer efficiently working. That's how that look a CSTR in the first figure over there. Okay. Then you have a system called upflow anaerobic sludge blanket reactor, UASB. The bacteria are itself form granules, so that's because sludge blanket. So you hold your SRT high. The bacteria easily doesn't get washed out. So the SRT will be high in UASB. Okay. And with a simple system, you can have a covered lagoon. Basically, you just put your wastewater in a pond, cover with a lagoon system. The gas is collected. You can use the gas collection. So these are some anaerobic digestive designs. Okay, next one. So when you do a design component, so you have to have a waste coming in and waste going out, holding in the digester. And then sometimes you need to excess waste that goes out, recirculated, digested sludge is taken out. So this is schematically, that's our basic component of an anaerobic digester. Okay, so if it is a continuously operating system, your input of waste coming continuously, you're holding in the bioreactor, bacteria doing all this job we talked about, producing methane, CO2 as the end product. And then the waste is going out, we call that effluent, and then the digester sludge is taken out and put in land application for biofertilizer. Okay. Next one. The excess biomass disposal is, if you have too much biomass, you can, accumulate as a sludge. You have to separate them, and you need to de-water the sludge, and then dispose them for beneficial use. Sometimes that could be used as a garden potting soil, but you need to de-water the sludge and make the soil separated. Okay, the sludge separated from water. Okay. Let's look at some of the more design of bioreactor. Okay, next one. These are the oldest design of anaerobic reactor, really old, in 1881, 1895, 1905. We call that Morris automatic scavenger, septic tank in your, if you use septic tank in your household, Imhoff tank. They are all operated under the same principle of anaerobic digestion. Okay, so you just hold your waste that come from your house, and that produce methane, and the water is let out after your carbon is removed, it goes out. And this is oldest bioreactor, IMHAF tank, septic tank. Okay, next one. Next one. The anaerobic digestion process really kicked up in Western European countries and the US when there was an oil crisis in the 1970s. They rapidly built up a lot of digesters everywhere and produced a lot of methane and, in the US. And also Western European countries. Then the oil embargo was taken out by the Middle Eastern country, then gasoline price got cheaper, then anaerobic digestion process fizzled out. But now there are some operated in big farming community. We don't have that many in the U.S. Okay, just to show you historically, again depends on your gasoline price. If gas price is high, this alternative will take off. If gas price is low, we lose interest on it. Okay, next one. Then you have the comparing. If you're doing your wastewater treatment, combining with aerobic, anaerobic system, you separate the solids that go to anaerobic digester. That's what's happening. You have the primary, secondary, tertiary treatment, and the secondary solids and primary solids that come from your sedimentation tank that goes into your anaerobic digester. Generally, that's what it is. The primary solid sludge is 35%, secondary is 25%. The total solid that go in the digester is 60% from your wastewater process. Okay, so if you have both aerobic and anaerobic system, they take the solids that goes into the digester. Okay. Again, talking about anaerobic treatment of solids here, the high solids basically animal manure, biological sludge we call night soil, and those are all common substrate used in anaerobic digester. And you have to design based on how much volatile solid in your system, how much SRT you need to put in. So your substrate varies from manure to human waste. Anaerobic digester can be used. Okay, next. All right, next one. So how do you achieve SRT? You need to play around with the substrate and play around with how much bacteria is retained in your bioreactor, and that's how you maintain your solid or sludge retention time. Okay. All right. So when you look at the waste treatment, the anaerobic treatment is a biological process carried out in the absence of oxygen. Oxygen is in aerobic system is your electron acceptor. So in the anaerobic system, for stabilization of organic material, conversion of organic material to methane, and the inorganic compound is such as CO2, ammonia is produced. So the simple reaction is organic material plus nutrient, anaerobic microbes produce methane, CO2, ammonia, and biomass. In the anaerobic process, there are two kinds. One is anaerobic fermentation. That's what we talked about so far. There is no external electron acceptor. Everything is fermentation reaction. All the electrons are put in the organic compound. Anaerobic respiration is if you have electron acceptor other than oxygen, like sulfate, nitrate, iron, manganese. The bacteria can use those as electron acceptor, and that we call anaerobic respiration. So there is a difference between fermentation and anaerobic respiration. Next slide. So anaerobic fermentation is, there is no electron acceptor in the system. So sulfate reducing bacteria need electron acceptor, so sulfate is your electron acceptor. So that's what the difference showing what is an anaerobic fermentation and anaerobic respiration, to show from glucose to ethanol, yesterday we talked about, that is fermentation. There is no external electron acceptor. Okay? In anaerobic respiration, you have electron acceptor like sulfate, carbon dioxide, nitrate are used as electron. All the hydrogen that's broken down from the substrate is dumped on to these electron acceptors. So that's why sulfate become hydrogen sulfide. Okay, you have nitrogen produced, and CO2 is your electron acceptor for methanogens and produce methane. Okay, next one. And the order of electron acceptor efficiency is oxygen is the best electron acceptor, followed by nitrate, sulfate, and carbon dioxide. That's the order of electron acceptor in the redox potential of electron transfer. So again, to go through the quickly, based on advantage of anaerobic process, the less energy requirement, there is no aeration, energy generation in the form of methane gas, less biomass generation. This is what's happening. If you use aerobic process for your waste, your soluble BOD is converted 50% to carbon dioxide and water, 50% to make new biomass, new bacteria in your system. Okay? We call volatile suspended solids. If you are using anaerobic process, if you have 1 kilogram of COD, 90% of it is going to methane and other gas. Only 10% is used to make new bacteria as biomass. So you can see most of them as the end product of your gas when you use anaerobic process. In aerobic process, 50-50, bacteria use 50% of the carbon to make more cell. So you have more biomass accumulation in your system. In anaerobic, you have less biomass accumulation. Okay, next slide. Other advantage of anaerobic is less nutrient requirement. You don't have to put a lot of nitrogen, phosphorus compared to aerobic system. Application of high organic loading rate is possible in anaerobic system. You can save some space when you use anaerobe. Ability to transform several hazardous solvents into end product like chloroform, trichloroethylene. Anaerobes can take those easily compared to aerobic bacteria. So they have high tolerance for some of those solvents. They can break it down. Okay? But it also has some limitations. This is the limitation for anaerobic digestion is that it takes a long time to start up. In initial startup for anaerobic process 30 to 40 days or sometimes 60 days to have all these bacteria to establish in your reactor. Okay? And if there is an upset in your reactor like a sour I told you are stuck in the system, it takes a long time to recover. Okay? Then there is some specific nutrient requirements that are some certain trace elements are needed for anaerobe like molybdenum. They need really in trace element. And they are more susceptible to environmental conditions. So those are some limitations of anaerobic process. Next one. Some more limitation, if you have too much sulfate, I told you hydrogen sulfide will, sulfur reducing bacteria will going to dominate. Effluent quality of treated wastewater also make a difference. So you have problem that also you have to take care of. Treatment of high protein and nitrogen containing wastewater is also an issue in anaerobic digester. So that you have to take a denitrification reaction to work with. Okay. So if you compare, if your wastewater you want to do it aerobic route, aerobic route, here is the pros and cons of aerobic and anaerobic system. Okay, anaerobic system, organic loading rate is high. Aerobic system, organic loading rate is low. Biomass yield, 10% is only going to biomass in anaerobic, 50% is going to aerobic. Okay, and specific substrate utilization, the high rate in anaerobic and low rate in aerobic. Startup time is long for anaerobe and less for aerobe. So there are some pros and cons on anaerobe and aerobic system. Some more SRT, longer SRT for anaerobe, shorter SRT for aerobic system. Microbiology is more complex in anaerobic. In aerobic system is simple microbes, not too many diversity there to worry about. Environmental process also highly susceptible to change in anaerobe, and aerobe can handle a little better. Okay? So the essential condition for anaerobic treatment, those are the one, repeating it again. We talked about you have to avoid oxygen, avoid toxicity, make sure there is enough alkalinity in the system. You need to have a nitrogen phosphorus ratio and trace element put in the system. Find out right SRT and HRT. Okay. So best wastewater that can be used for your energies is alcohol distillery wastewater, brewery and winery wastewater, sugar processing wastewater, starch manufacturing, food processing, bakery plant, pulp and paper mill, dairy product, slaughterhouse, petrochemical waste. These are all good substrate for your anaerobic digester operation. Okay. So we have two kinds of wastewater, the low rate and high rate. Low rates are these anaerobic pond, septic tank, Imhoff tank we talked about. The high rate are the recent modern reactor. Those are your anaerobic sludge blanket reactor, sequencing batch reactor, anaerobic filter, and anaerobic baffle reactor. These are all little more sophisticated bioreactor that can handle high loading rate of your substrate. Okay. The simple process is your anaerobic contact process. That's basically a tank. Make it anaerobic and put your waste in there. Bacteria contact with the substrate and produce your methane gas. It's a low maintenance, but the efficiency is not very high. But if you have a small amount of wastewater, this will work for you. Okay. Next one. That's how ACP work. Anaerobic contact process is relatively for dilute wastewater, not too much complex substrate involved. Biomass concentration range from 4 to 6 gram per liter and is low COD, and this process works for such small concentration of waste. Okay. Then we have anaerobic filter, first discovered by Dr. McCarty in the 1960s. And basically what this bioreactor is, you put some filter media in the tank, and the filter media is to hold your bacteria and to have long SRT. So when you continuously pump wastewater, the bacteria is trapped around this filter media. It could be plastic media or rock or pebble or ceramic tiles. You fill them up in the bioreactor, so you can have long solid retention time. You're not going to lose the bacteria out of the system. So that's called anaerobic filter system. Next one. And that's how the system works. You can operate upflow or downflow. You pump your wastewater upflow or by gravity downflow, but you are filling them with some kind of filter to hold your bacteria. Okay, next one. So the packing materials are anything like that. This is what you put inside the filter system. It's be different kinds of plastic. The purpose is to hold bacteria inside long time. The bacteria form biofilm on the plastic filter, so you don't lose bacteria. That's the purpose of filter. Okay, you have a long SRT. Next one. So those are some of the advantages of using anaerobic filters. It could be operated either downflow mode or upflow mode, depending on how you want to use them. Okay. That's upflow. You're pumping it up, and the downflow is through gravity. Then the next sophisticated one is upflow anaerobic sludge blanket reactor, developed by Dr. Ledinga in Wageningen University, Netherlands, in the 70s. So what the disadvantage of filter is, you have less wide volume, less empty space, because you're putting a lot of filter media. You're treating less wastewater, right? Because your tank is filled with your filter. To avoid that, this reactor come up with this plan. What it does is it creates all the bacteria to form a big clump, and the bacteria, we call that granules. The bacteria itself form big chunk. You don't have to have any filter media in your tank, and all the bacteria are in this form of granule, and they have a high settling velocity. They have density is high, so they settle so quickly, so they don't float outside the reactor. So you hold your bacteria for a long period of time, and you have a lot of space in the bioreactor to treat your wastewater. Okay, next one. Then we have another design in Iowa State they produce, and they call static granular bed reactor, and this is just the opposite of upflow sludge blanket reactor. Here it is a downflow system. So here you have bacteria are stayed at the bottom, but you're pumping it from downflow. It's a simple operation with few moving parts, and the major issue is for when you operate continuous for a long period of time, you have head loss due to buildup of solids in this type of reactor. Okay. Next one. That's how the reactor look like. Okay. And we just talked about before, sulfate has some problem. If you have sulfate, the sulfate reducing bacteria are going to compete with your substrate, so your methane production will go down. So that's the reaction of how sulfate reducing bacteria compete with the carbon, the COD, and produce hydrogen sulfide. Okay, so that's their reaction of how sulfate reducing bacteria work, how much electrons are used, and how much sulfate is used as the electron acceptor to produce hydrogen sulfide. If you do the stoichiometric analysis, theoretically 1 gram of COD is needed to reduce 1.5 gram of sulfate. So too much. If you allow a sulfate, for every 1 gram of COD, 1.5 gram of sulfate is reduced. Okay, if you work out the calculation. So if you have too much sulfate in your wastewater, you can kind of reduce your sulfate going into your system based on these calculations. Okay, next slide. Another type of reactor, expanded bed bioreactor, and in this case, the bioreactor is kind of slowly expand, and there is no sludge blanket, there is no filter here. The bioreactor itself is expanded bed bioreactor. Okay, next slide. So that's how the expanded bed bioreactor look like. So you have a bed at the 1/3 volume of the bioreactor. It slowly expands up and down based on your operation of your bioreactor. Next one. Another kind is called fluidized bed bioreactor. It is filled with 1/3 volume by sand particles, and when you pump the wastewater from upflow, the sand create a fluidized motion. So you don't have any mixing involved. The bacteria are trapped around the sand particles, and each particle have its own fluidized motion. So you don't have to mix the system. So mass transfer takes place by pumping the waste, moving the sand bed in a fluidized motion. And that's why the fluidized sand particle look like. That's another bioreactor. Okay. And then we have hybrid system. You combine 2 bioreactor in one. In this case, you combine with your upflow sludge blanket with a filter. So you can modify your bioreactor by putting 2 systems in one based on what type of wastewater you're using. If you look at all this system, what they're doing is they're holding your SRT long time. That's the major theme of this bioreactor. You wanted to make your bacteria retained inside by putting some kind of trap, some filter, sludge blanket, sand particles. That's what they're doing here. Okay, next one. Another design is called anaerobic baffle reactor. You have series of baffle, down baffle, up baffle. The wastewater goes through like a snake. It just moves up and down, up and down. So you hold your bacteria, your sludge inside for a long time. Right? That is called anaerobic baffle reactor. Another design out there, commercially used. And then you can use your reactor depending on what kind of waste coming in. We call that sequencing batch reactor. You can operate sequentially, and in this case, you get the wastewater come in, settle, allow to react with bacteria, and once it's done, you decant and bring in fresh batch. It's a batch reactor. If you don't have too much wastewater, it's a seasonal operation. This sequencing batch reactor will work for you. So instead of continuous reactor, you're using batch in a sequential manner. That's why it's called SBR, sequencing batch reactor. Okay. And again, for all this reactor, you need to determine your organic volumetric loading or OLR That you have to play around with it to conduct a pilot scale study, find out removal efficiency of your system, and select the right organic loading. To do that, you need a flow rate, you need a COD concentration of substrate, you need your volume of bioreactor. So you can figure out how much COD you want to put in. Okay, next one. All right. So what happens to the leftover? So you produce your gas, then you produce solids inside. So common misconception is anaerobic digester in the digester the biogas produce it suck up the nutrient. It's not the case. So you have a lot of leftover nitrogen phosphorus in the system, and those are called digestate, and digestate is a good fertilizer. You could use it in biofertilizer apply in your land for replacing your commercial fertilizer. So that's what's happening in the digestate. Okay. Next one. So I'm going to continue until 1, so I hope you guys are still with me. Okay. So I went through microbial process, various bacteria involved, environmental factors, and how to design bioreactor. Now we're going to talk about digestate. So what happened? Yeah. You want a break? All right. You want to take 5 minutes break, you announce? Okay. It's too much time to hold you. You need a brain break, so let's take a 5-minute break. Okay. I'll come back. Okay. So I told you I'll take the time from previous lecture. Okay. Yeah. Let's take a break. Okay.
After 5 minutes, enough minutes, 5 minutes.
All right. Welcome back. So we are going to finish up this anaerobic digester talk. So we talked about some fundamentals of microbes, environmental factor, reactor design. Now we're going to talk about the digestate. What happens, the material that we call sludge or digestate after the digestion process. Okay. So here is the overview of what is happening in your anaerobic digester. So you have input that going in, and bacteria doing the reaction, you get your final product of methane, and then you have the material that is inside, we call that digestate or sludge, and then you have to take care of that. And then the more complicated one, the gas, and the gas you need to purify if it is hydrogen sulfide. If it is too much, we'll continue in the afternoon. I have that part also, how to purify gas. Okay. Next slide. Yeah, go down next slide. Yeah. So the biogas, what is in it? You have methane, we have carbon dioxide, nitrogen, a little bit of oxygen, hydrogen sulfide, ammonia. Those are all the component of your final product of digesting your waste. Okay. So this gas can be used as a transportation fuel with a methane content of 95 to 99%. That's why you have to purify your gas, remove other impurities. You only have methane, so you could use that methane to run your car. All right. So that could be used as a transportation fuel. Next one. So very quickly, we're going to go through how we're going to clean up the biogas. We have a separation of gas component. You want to remove your moisture in the gas, remove sulfide, remove ammonia. And when you do that, we can pump the gas straight into your generator, and you can produce electricity. Okay, you can use the electricity generation. Then we can also upgrade your biogas by further separation of your CO2 with biogas and purify your gas up to 98% methane by volume. So biogas can be upgraded and linked to your compressed natural gas, CNG. So you can sell the biogas into your gas company. They can buy it. In the US, that's what they do. You purify your gas and they will pay you to get your gas if you're operating a lot of anaerobic digester. Okay. So the biogas is impurities. You have this, these are the components you need to remove before you can sell your gas to either a gas company or run electricity. Okay. So the carbon dioxide is not necessarily impurity, it will dilute your gas, so you need to have problem with power generation. So You can do various method of absorption, adsorption, cryogen. Those are the method out there. If you look at this graph, it shows the efficiency of removal of carbon dioxide in various methods. These are all commercially operated successfully method to remove your CO2. Okay, so using various absorbent to absorb your CO2, and you're also using cryogen to remove CO2. Okay. You can also use pressure water scrubber or gas washing system. The gas is passed through this absorption system. So the only thing that comes out is CO2. Okay, I mean methane. CO2 is absorbed. Okay. So it also simultaneously removes your hydrogen sulfide. Any moisture is also removed. It's very efficient. And depends on the water quality Henry's laws. You can calculate and make your gas as pure as possible, 98 to 99% methane in the system. Okay. Moisture is a little bit of moisture in the gas. You need to remove that. It's saturated with water, so you have to use the condensation cooler pipe section. You put a trap in your gas collection system. Water is collected in that trap, gas is passed on. All right, so you can remove moisture using this kind of system by condensation. Condensed water accumulates at the pipe at the bottom, hinder the gas flow, pressure built up in the digester. But you can pumped up the gas, but trap the water. Okay. Cooling the biogas to condense the water, and then you remove the condensed water. That's how the system look like. You can trap and get the moisture out. Okay. The hydrogen sulfide is another issue. How you're going to remove it? If you don't remove it, it's going to corrode your pipe and make a problem. So, so if you have too much sulfate in your wastewater, you have a lot of hydrogen sulfide. And so the wastewater that has a lot of sulfate is listed there. Molasses, tannery, distillery waste, they all have a lot of sulfate in it. You look at the concentration, 2,000 to 14,000, lot of sulfate, that you have a lot of hydrogen sulfide production. If you have H2S, that's what happened in your piping inside your digester. It will corrode because H2S is a corrosive gas, and so you got to try to eliminate H2S by putting H2S scrubber. You can get the H2S absorbed. Okay. So sulfide limits various application because we have too much sulfide. It won't run your turbine engine. It's going to create problem, combustion issues. It also has the vehicular fuel operation is inefficient, and also has affected natural gas pipeline. So, so it has a high maintenance cost if you have high hydrogen sulfide. So there are a lot of methods out there. You can use physical method like water scrubbing, pressure swing adsorption. You can also chemical method. You can use iron sponge. Iron sponge can absorb your hydrogen sulfide. You can also use biological method. You can use chemoautotrophic bacteria that can oxidize your hydrogen sulfide back to sulfate. Okay. Then you can also use biofilter, direct addition of air to dilute your hydrogen sulfide. Okay. That's how an iron sponge look like. When you pass the gas through it, the hydrogen sulfide is all collected in that iron. Iron absorbs your hydrogen sulfide, and the gas that passed through is without hydrogen sulfide. Okay. We can also have direct injection, air injection in the headspace to dilute your hydrogen sulfide. That's another way to do that. Okay. Next one. Another thing most people don't know is the gas also contains this siloxane because you have silica in your waste stream. If you have silica in it, you produce siloxane. These are organic silicon polymer used in your personal care product like shampoos and conditioners and deodorants. And they in the digester they produce this siloxane, and you have to it's a volatile gas, and you have to remove them most of the time. All right, this is another issue. Okay. And other thing we have is digestate. The digestate is all the organic material that after gas methane is produced, you left with a sludge. That sludge looks like this. The digestate can be pelletized in the pellet in a high density form, or you can use the sludge and pump it back into the farm as a natural fertilizer. Next one. That's a general characteristic of digestate. You can look at the different parameters of what is left in the digestate: nitrogen, phosphorus, ammonia, the values that range most of the time for different ways, and then absolute E values for each of them. Okay, next one. Nutrient content of the various waste, whether it is digested cow slurry, digested pig slurry, you can see that the whole thing, that is what is in the digestate: dry matter, total nitrogen, available nitrogen, organic carbon, phosphate. So depending on your waste going in the system, you have all these in the components in your digestate. Okay. Next one. If it is a livestock slurry, if there is too much heavy metal in it, you need to worry about heavy metal accumulation like copper, cadmium, chromium, mercury. You don't want that in your farm. So most of the time you can see the permissible limit at the last column. You don't have that issue at all. Most of the time all these heavy metals are within the permissible limit, so we don't have that. So EPA has that limit. If it is exceeding that, you cannot apply in your farm, okay, that heavy metal. How do you process the digestate? You can separate solid liquid separation, use a screen, you can use a sludge press, and also you can centrifuge depending on volume of sludge you produce. Okay. So you can use the various screen, we can use various presses, and if there's too much water in your sludge, you can use centrifuge systems. Okay. So the effluent can be used as a fertilizer with the irrigation channel, drip irrigation, spraying in, or you can produce true white. True white is like a pellet that you can apply as a fertilizer. You can use this waste into your algae production, algal pond. So that's an example of how effluent is used. In the US, commonly used as fertigation. So you can, go over in a tractor and put this sludge into the farm in form of irrigation system slowly into the soil. Okay. You can see the effluent, compare how they are used in the drip irrigation. You can pump the sludge in the pipe into the farm, and it's really good application value for it. Okay. And these are some of the equipment they use to put this sludge in the farm, and high, because in the US the average farm is 1,500 acres. They can afford to use big machinery like this. Okay, that's how that machinery looks like. So if you look at what it does, it's rich in nitrogen, and the nitrogen are a usable form. Easily plant can uptake this nitrogen. Okay, so high in nitrogen, high in phosphorus. So you can use this for agriculture as your biofertilizer. Okay, next one. You can use commercially. You can take your sludge and produce this true white, effluent sludge from the anaerobic digester, which is the ammonium magnesium phosphate. It comes in this pellet, and this pellet you can apply as your fertilizer. Okay, we call it true white, and that is produced from anaerobic digester sludge. And then you can use the sludge in your algal pond to produce algae, because there's a lot of nutrients still there. So algae can easily grow. It's an example of algal cultivation using anaerobic digester digestate sludge. Okay. And that's how a solid fraction look like when you try to get the sludge out. It really come out really large quantity if it is a big bioreactor. Okay. And you can dry it out and use in different form, whatever way you want it. So you need to de-water them, remove the water, and you have a dry solid used as biofertilizer. You can also use this when it is dry with no water. You can use as animal bed bedding in the, when you have animal husbandry, you can do composting, you can use fuel pellets, you can make it a pellet and burn it as a furnace, and also you can use the fiber for various purposes. You can make a fiber board out of it, and I will show you a picture of it. Okay. You can ask these are the bedding we use from the digestate. When you remove the water, it could be used as a bedding. Okay, next one. Compost, you can compost it and sell it commercial as compost. Okay. That's how the pellets look like. Pellet operating machine, and pelletize it. That pellet you can burn easily, like your firewood. You can burn it like a coal, and you can use it in a commercial scale in your furnace operation in a big wherever you need heat. Okay. That's using the same digestate. After you remove the water, you mix with plastic and you make various mat out of that. Okay, that's another use. Fiber board you can use them. Let's move next one. Next slide. That's overview of. Okay, next slide. We have to think this holistically. You are using anaerobic digester as a biorefinery. Nothing is going to waste. Okay, you get your raw material, produce your main value-added product is your methane and sometimes hydrogen, and then you are using digestate to make all this product. Right, the solid is produced, and if you look at it holistically, it's a biorefinery. What do you need? You tailor them for it. If you need fertilizer, if you need bedding, if you need fiberboard, you can use them, all of them. So it's more like a biorefinery concept you can operate anaerobic digester. Next slide. So if you look at the anaerobic digestion process, United Nations and world bodies, they really like anaerobic digestion technology. It's an old technology with a lot of advancement with the recent research. It's a win-win technology, ready to go, ready to scale up. Most of the kinks are worked out. All those problems most of the time already worked out. Anaerobic digestion is the main part of the circular economy concept. So United Nations really favor anaerobic digestion technology. Okay? And if you look at household, in India we use biogas, we call that gobar gas in some of the villages, and that's how the household anaerobic digestion biorefinery works. You get your gas, you get the digestate for your fertilizer use. Okay? Next one. Next one. So I have another 10 minutes to finish up, otherwise I will extend this. Okay, so this is an advancement in anaerobic digestion. So I just want to talk about, so when you have sour, we talked about, you're stuck with anaerobic digestion because of you create too much fatty acid, right? So the one group of bacteria dominating, so methanogen is inhibited. So how do you solve that problem? That problem is by microaeration. So you can pump oxygen into your anaerobic digester. So the hydrolysis process will complete and convert them to acetate. Okay, so that is called microaeration to remove your sour or stuck process in anaerobic digestion. So what you are doing is pumping a little bit of oxygen. So it doesn't kill all the bacteria, it removes the accumulated fatty acid in your bioreactor. Okay? So at a pulsing system is how they use the microaeration pulse, and small quantity of oxygen is applied. Okay? Next. So when you do that, when the sour system has a lot of fatty acid, and when you apply this microaeration, your fatty acid goes down. So you correct the system, so the methanogen will come back, and so you correct your problem. So that's how the microaeration works. So it to solve the problem of fatty acid accumulation. Next slide. So microaeration is a natural phenomenon. All you need is think about cow, how cow does it. Okay, the cow eats the grass, and then it goes sit there and regurgitate, right? It brings the material back in there and it chews it up. It takes some oxygen with it. We are just exactly copying that in the anaerobic digester. Okay, next slide. So that's what that's showing that. So grass-eating lignocellulosic cow, rumen digestion, it brings that ruminant back into the mouth and chew it back, and along with that the air goes in that. That is microaeration. It's a natural phenomenon. You're not killing your methanogens. Okay, so this is what your microaeration does to your anaerobic digester. Okay, so to understand this concept of anaerobic process, your redox conditions is important. So that's a redox scale on top. Anything above 0 is aerobic, negative condition, negative millivolt is anaerobic, and microaerobic is really right in between. So you pulse that microaerobic condition, so you're not basically affecting anaerobes. That anaerobes are not completely died off. Okay, you take care of the accumulation of fatty acids. Okay, next one. This showing you when you pulse the microaeration, that's what your redox potential is inside the anaerobic digester. There is not a big change. It falls within that negative millivolt range. Okay, next one. We did that experiment and with my colleague Dr. Kanal, and that's the data showing that the microaeration will solve your sour situation of when the bacteria are not in concert, one system take over another. We can pulse oxygen in small dose that will clear up the problem. Okay. Just to show you this how the reactor system was set up. Okay, next one. That's how your lab reactor looks like. So we use co-digestion. We use cow manure with all this different substrate of lignocellulosic mixed in a different ratio, and we are producing methane, and by solving microaeration problem by solving sour system. Okay, next one. There's just data as just to show you how microaeration works. Okay, next, continue that. So you can see that microaerobic condition takes care of your volatile fatty acid accumulation, and your methane yield is going to go up. It's not killing your methanogenic bacteria. Okay. When you have a sour situation, volatile fatty acids accumulate. When you pulse the oxygen, volatile acid come down. So that's what you're doing. So what happens when you pulse oxygen? There are a lot of facultative bacteria that multiply faster and consume these fatty acids and make acetic acid, which is what we want. Okay. Just experimentally showing this process again. We repeated it. Okay. And we show the reduction of volatile fatty acid by pulsing small amount of oxygen. Okay. Methane yield went up. Okay. So just show you after we pump oxygen, next slide, we don't have any problem of microbial variation. Bacteria remains the same. We did the analysis of microaerobic versus anaerobic condition. There is not a big change. All the bacteria, all of them are still there, but different quantity. Okay. And just to show you the list of bacteria when you apply microaerobic condition, what are they? And they're listed over here. These are the research done, and just to show you, microaerobic condition will take care of your fatty acid accumulation in anaerobic digester. Okay. Also, microaeration did not increase your CO2. Your methane went up, which is surprising. Okay. And your methanogenic bacterial system did not change at all. When the general concept is when you have oxygen, your methanogen will die. Right? In this case, methanogen did not die because we are not putting too much oxygen. We are putting less than 1%. So that is called micropulsing. Okay. Next slide. So basically, microaeration enhanced hydrogenotrophic methanogen. It really works this system. So this is the new advancement in anaerobic digester. We are pumping a little bit of oxygen to solve a problem in the digester. Okay. Next slide. And just to show you biochemically what is happening when you use the nano amount of aerobic pumping into the digester, biochemically what is happening? The facultative bacteria take over, consume your fatty acids, and bring down your acids to acceptable conditions. The methanogens is not inhibited. Okay. So summary, the nanoaerobic digestion summary, it really works. It takes care of your fatty acid accumulation problem. Methanogens came back. Your digester is not sour anymore, and it is working really as it was before it was affected. Okay. Next slide. So in conclusion, your OPR, which is your redox potential based microreaction, promoted facultative bacteria. It take care of your VFA concentration, create a stable anaerobic digestion for your organic loading to solve this new problem of sour or stuck in the anaerobic digester. Next slide. So I just want to conclude that some of the work was credited with Dr. Samir Kanal from Insta Hawaii and Norwegian University Life Sciences, Dr. Jensen. Okay, collaboration with these people. Okay, thanks very much. And I think I took 2 hour time. I hope you guys are okay. All right, let's take a break and they're done. Yeah, thank you.